Thank you for inviting me to this event. Of course, I would love to be in Spain and 20 degrees, not here in Oslo with 0 degrees and snow. But when it's not possible, I'm glad that we can do this digitally. So my role here today, I have several roles, but primarily I will discuss how and why we need novel governance for novel gene technologies. And like Holger just said at the end of his talk, it's not just about the science, it's of course also a lot about the politics. So I will start by giving you a background or a rationale as to why we need to think new about regulation. Let's see. Yeah, there we go. So first of all, as you also heard from Holger, genetic engineering is not one thing. It's a wide range of possible changes that can be made with genetic engineering. Everything from temporary changes in gene expression and genetic mutations that mimic those that already exist or can arise in nature, through gene transfer and transgenics, all the way up to synthetic biology and fully synthetic organisms. And of course, these have very different opportunities and challenges associated with them. Of course, we know also that genetic engineering can be used to address many different challenges, and many of them are very important, especially in terms of sustainability. So genetic engineering can contribute to improved food security, productivity, environmental impact, animal welfare, biodiversity, and adaptation to and mitigation of climate change, for example. And although some like to say that consumer opinions are quite one-dimensional, I will disagree. And this is partly because of the report that Antonio mentioned that we have done in the Gene Innovate Consortium, where we have asked Norwegian consumers what they think about genome editing. And as we can see, the attitudes are very nuanced. So for example, they are very positive towards uses that contribute clearly to sustainability or that have consumer benefits, such as reducing the uses of pesticides in crops or improving the nutritional content, and also in livestock breeding, where improving animal health, for example, is something that most consumers were positive towards. They were, however, less positive and primarily negative towards using genome editing for what can maybe be conceived as more trivial traits, such as changing the color of a product. And they were also concerned about traits that could impact animal welfare, such as increased productivity in animals. So, but the main take-home message from this is that, of course, the opinion of the consumer depends on what the genome editing technology is used for. Despite all these important nuances, we see that the political debate is still very black and white, and it is very polarized. And this is, of course, reflected in the binary regulatory system that we have in the EU. As you probably all know, it's either a GMO or it's not a GMO. And this applies to all products. And of course, there were a long time, there was unclarity about how genome editing would be regulated. But after the Court of Justice expressed their opinion, it is clear that everything that is made with genetic engineering are by default defined as GMOs with the full set of regulation. But of course, this has consequences. So first of all, as I think most scientists will agree, that the current risk assessment regime for products developed with genetic engineering can be rather disproportionate, and especially when we talk about genome editing and other products that are similar to what can be made with traditional methods. And this is also something that was pointed out by the EU Commission's own scientific advice mechanism or committee. They had a report in 2017 where they had several conclusions. First of all, they said, and they stated that there are differences between new breeding technologies. Some of them are more like classic genetic modification, while others have similar or greater similarities with conventional breeding techniques. And therefore, this wide variation in new techniques means that grouping them would not necessarily be appropriate for scientific or other reasons. And when it comes to off-target effects, which Holger has elegantly explained already, off-target effects producing or in organisms produced with new techniques is much lower than the off-target effects with conventional breeding and classic genetic modification. And they also said that genetically and phenotypically similar products developed with different methods are not expected to involve different risks. So, and of course, in the regulatory system, they are treated as one group and they are treated that genetic engineering products are more risky inherently than other products, which is not necessarily scientifically correct. Of course, the current regulation also means that there is a very time-consuming and expensive approval process, which means that marketing authorization would be unattainable for many academic groups and small businesses. And of course, the current regulatory situation in Europe gives a competitive disadvantage and geopolitical imbalance. If we look at the global regulation, we can see that many of the different countries and regions internationally have decided to take a more lenient approach to genome editing. And here the exception is Europe and New Zealand, which have decided to regulate genome edited products as GMOs. And of course, this has implications. If we look at the patent landscape, we can see that the number of patented patents in the genome or CRISPR technology itself, also within medical applications, industrial applications, and also in plant and animal breeding, we can see that way ahead of the pack are, of course, the US and China in this case. And Europe is trailing quite far behind. And this is, in the opinion of many, especially scientists, due to the regulatory situation. And of course, we also see that governments across the globe are investing in this technology quite a lot, and not just the usual suspects, such as the US and South America, but also, for example, China, which have made a deal with one of the pig breeding companies to develop disease-resistant pigs, and also Russia, who have invested quite a lot of money in plant genome editing programs. And for those who work with plant and animal breeding in Europe, this is a concern. And this is the head of research and development at Norsvin, who do pig breeding. And they are quite concerned, and especially that one of their competitors have succeeded in making genome edited disease-resistant pigs that in principle can result in Norsvin losing a lot of their business, because they are an international company who sell genetics to the international market. There are other consequences also. With the current heavy regulation, producers will choose inferior non-GM methods to achieve similar results. And one very interesting example is this genetically engineered microbe that has been developed in the US by researchers at MIT. These microbes can fix nitrogen for maize plants, which is not something that occurs normally in nature, but it mimics what happens with other types of plants, such as pea plants, that have this ability. So this reduces the need for artificial fertilizer, which again reduces the nitrogen runoff to the environment, and also emissions of the greenhouse gas nitrous oxide, and it significantly increases the yield. And this is not regulated as GMO in the US, and it's used in fields across the country. And when I asked the researchers if this is something that could potentially also be aimed for the European market, they said that no, they will not, because the regulatory burden is too high. But they will try to make similar microbes using mutagenesis, because of course that is not regulated as GMO. But of course it will not be as precise as the genetically engineered microbes, so it might take a while before this is ready for the European market. And another example is from here in Norway, where researchers at the Marine Institute are developing CRISPR salmon. They knock out a gene that results in the salmon being germ cell free, so they cannot reproduce. And of course, the main benefit of this is that these salmon cannot interbreed with wild salmon if they escape. And they also have improved health, actually. But this is then, because they are genome edited, they are classified as GMO, which means that the industry rather will choose a different technology. They will choose something called triploidization, where they expose the developing eggs to high pressure, which results in them becoming triploid. They have an extra set of chromosomes, which renders them sterile and therefore achieves basically the same result. But of course, this is not necessarily as good for the animals. They are more temperature sensitive and they have a higher mortality than other salmon. So that is an unfortunate consequence of the current regulation. And also different regulations and labeling requirements for identical products will mean that the industry interest will be lower. And which Holger also briefly mentioned, that enforcing the legislation will be challenging. So actually the European Commission's own technical experts, the JRC, they published a report a couple of years ago where they pointed out some challenges in terms of detection of genetically modified food or feed originating from genome editing. And the main conclusions were that detection is really only possible when you have information about the genetic alteration beforehand, because the mutations themselves are indistinguishable from what's going to occur in nature or be achieved with traditional breeding. And this means that enforcing the current requirements for detection and traceability will be difficult. And that, again, of course, will mean that it could be difficult to clear food and feed imports arriving at the EU, because they will not be classified as GMO in the country of origin, which means that it's up to Europe to prove that they are GMOs. And this can really take time. And of course, there's the World Trade Organization. We have very strict trade rules, which I'm quite sure that in the coming years, when genome edited products are market ready, we will see some disputes over the trade situation if Europe will deny access to genome edited products. So that's the consequences of the current regulation. But also there are consequences of completely deregulating genome edited products, which I know that many, at least scientists, are advocating. First of all, you will have no oversight over how the technology is used. And there was an example in Brazil, not last year, but a couple of years ago, where you have genome edited cattle that are hornless or polled, as they're called, which were not regulated, but that by accident were discovered by the FDA in the US that they actually had an integration event of the bacterial DNA that was used for the transformation event that had snuck into the cattle DNA. And this really shows that it's not always predictable what happens and that regulatory overview is an advantage. And if you don't have a regulation, there is no option to assess the risk or other consequences, such as impact on sustainability. And again, I will refer to the scientific advice mechanism report, where they also stated that in principle, no conclusions can be made concerning the absolute or comparative risks between different techniques, and that it can only really be done on a case-by-case basis, and it will depend on the characteristics of the end product. And also from our survey, when we asked the public, even though they were positive towards several users of genome editing. We asked if they were worried about risk, and the majority were very or somewhat worried about health risk or and environmental risk. And regardless of whether or not this is correct and there is an actual risk, the consumers need reassurance, and regulatory oversight may provide such reassurance. And then finally, no labeling. With this, if you deregulate, you really have no option of labeling, which means you really don't have a consumer choice. And again, I will refer to the survey that we did. We asked how important or unimportant it might be for them that genome editing products are labeled. And a clear majority said that it is very or somewhat important that the products are labeled. But as opposed to the current system, they clearly also want the label to distinguish between genome editing and classical GMO, and it should contain information about the trait that has been changed and the purpose for making it, because this is relevant for the consumer, as we saw earlier. So what is needed? We need an appropriate regulatory framework that really shows innovation and governance. We need openness and transparency. And we need a knowledge-based and goal-oriented and inclusive public dialogue. So the main question to ask is how we can utilize the potential of genetic engineering in a safe and sustainable way that also promotes trust and transparency, and that can feasibly be implemented in the current political climate in the EU, because that is not an easy situation. So that is the basis for the work that I've been involved in with the Norwegian Biotechnology Advisory Board a few years ago, where we developed a novel governance framework for GMO. And because of the feasibility aspect, we took the current EU framework as a starting point, but tried to develop it to be more updated to the current situation. So in brief, we recommended that organisms that don't have heritable genetic changes, if you have just manipulated gene expression, for example, this should not be regulated. But other organisms should. If you have made edits to the genome, they should be regulated in some way, but it can be differentiated and tiered. So on the lowest tier, we propose to have genetically engineered organisms that have changes that either exist or can arise naturally and be achieved using conventional breeding methods. And for this, we only suggest a notification. And typically, this would be products such as the one that Holger mentioned that Kasia Gao are developing with mildew-resistant plants that have a mutation in one gene that renders them resistant to infection. This is typically something that would be on tier 1. Another example is something that you could achieve with crossbreeding, such as the polled cattle, where if you, instead of crossing them and back crossing to get rid of everything else, you can do the genome edit to get the polled offspring. On the second tier, we propose that there should be organisms that have other species-specific genetic changes, which means basically larger changes, but that don't cross species barriers, which would be subject to an expedited assessment and approval. And this, in our opinion, would typically be something along the lines of what Holger explained with chromosome engineering. So for example, deleting a large part of a chromosome, which there has been examples of in rice, for example. This could maybe be appropriate for the tier 2. And then finally on tier 3, there are organisms that have genetic changes that either cross species barriers, which are basically today's GMOs, or that involve synthetic or artificial DNA sequences, and they should be subject to standard assessment and approval. This would then, like I said, typically be a GMO, a plant that has a gene from a bacterium inserted, or it could be more synthetic organisms. One example are researchers are working on synthetic bacteria that can change color and work as camouflage for military vehicles, or gene drives, which involves spreading out genetic changes in wild populations, such as gene drive mosquitoes to eradicate malaria. this should be strictly regulated, in our opinion. And importantly, what you can achieve with the sort of tiered system is that you can tailor labeling and traceability and detection requirements to each tier. So maybe you could exempt tier 1 from a detection requirement, and thereby solving one of the really big headaches of policymakers today. And also, what is already part of the Norwegian legislation is a focus on aspects such as societal benefits, sustainability, and ethics, which should be included in the assessment. And this way, you actually get, you allow for more weight to be placed on the benefits of a product, similar to how, for example, medicinal products are assessed today. And this way, we avoid the sort of one-dimensional focus on risk as the only relevant criterion for approval. So if you have a large benefit, that will weigh quite heavily. And to develop this, this proposal, we placed public dialogue at the heart of the process, because we believe that the public is vital for the future of genome editing. So we asked the Norwegian public what they thought about genetic engineering in general and regulation, and also our proposed regulatory model. And we got replies from over 50 Norwegian organizations who expressed their views. And on the one hand, typically academia and industry, for them, some of the really important aspects are they need an enabling framework that lowers the regulatory hurdle compared to how it is today. They also want something that is science-based and risk proportionate, and they want predictability. They want to know what they are facing when they are applying for approval. On the other side, we have typically environmental NGOs that express concerns that we have a lack of experience with these new technologies. They're concerned about the impact on ecosystems when the developments are occurring rapidly. And also they really want a need for precaution. And this is, of course, something that is very deeply ingrained in the European legislation. But even though there were differences in opinion, there were also many shared views between the different stakeholders. Most agreed that there is a value in regulatory oversight, and also that you need public trust. They agreed that gene editing and other genetic engineering technologies can contribute to sustainable agriculture and aquaculture. Everyone agreed that European competitiveness on the international market is crucial, and that it is important to protect the health and the environment and to promote societal benefits, sustainability, and ethics. And we believe that the regulatory model that we developed encompasses all these aspects. It is science-based, it lowers the regulatory hurdle for using genetic engineering while still providing oversight and control. And this in turn inspires transparency and public trust. And while it is difficult, in our opinion, to get political backing for a radical overhaul of the regulation in the current political climate, our model offers a compromise that hopefully there could be political willingness for. So we believe that all of these are safeguarded. And towards the end, I would like to just mention that recently there was a report published by the EU Commission's own ethics group about genome editing, and some of their recommendations regarding regulation were that you need a systems approach to the evaluation of costs and benefits, similar to what we have argued, including the wider impacts on ecosystems and agricultural, natural biodiversity, land use, economic impact, and food security. Regulation should be proportional to the risk, and they recommend a light touch regulation for example, gene silencing, or where the gene change could have been achieved naturally. But where the modification involves gene from non-sexually compatible organisms, there should be a detailed evaluation, including a requirement to test the new variety in field trials under different conditions. So in principle, it's arguing along the same lines that we do. And I will also, our proposal also answers the call for innovation that has been called for by others, such as the German Ethics Council, that also proposes a sort of differentiated tiered system And also the World Economic Forum has called for innovation in governance that is more inclusive and forward-thinking that the current system is. And also for example, the interest organization Fabre TP, which is an animal breeding organization in Europe, they have expressed that research on new breeding techniques like genome editing should be regulated but not prohibited by any regulation. Of course, there are also now a big push in Europe, for example, from a German Green fraction, interestingly, that are now pushing for an overhaul of the regulation. And many of you probably heard also that France's agriculture minister has called for different regulation of CRISPR crops that they aren't really GMOs. So now we are all waiting for the results of the European Commission study on the new genomic techniques, which is due this month. I'm very hopeful that they will really be forward-thinking and really give a good basis for policy development in the coming years. And also the legislation in Norway is under review because of these developments. I'm part of the committee that will give the recommendations on this and on how to make a future-oriented framework for genome editing. And I will just end by a couple of statements. So if we get it wrong by over-regulating these new technologies, will mean a huge opportunity costs. And it could result in a lack of transparency because genome edited products could in principle go undetected. You don't really have to declare it because you cannot prove that they are genome edited products. So if you want a transparency, you need to lower the regulatory hurdle. On the other hand, if you underregulate the products, that can result in a lack of control and a lack of trust. And we do really need the consumers on board for genetic engineering to have a real future. But getting it right will allow us to harness the potential of genome editing and other genetic engineering technologies to enable a transition to a green economy and a sustainable future in a safe way, while also maintaining public trust. I will end there and be happy to take questions. Thank you.